All right, folks, let's start. Last module, and we have 4 chapters. We're not going to prolong it. We're just going to finish it just one chapter at a day, because you will see that the intensity is a little bit less compared to that. I am not talking about too much about the exam. I will just post an afterthought, and if you have any questions, just let me know. Because unfortunately, these are some of the things that there are several different ways that I can track how many people are watching the videos at home. I'm not talking about the ones who are coming to the class, not coming to the class. And believe me, we have 66 people in the class. 50 people so far watched the final review. So, I mean, like 86% average is like, put me in a hot seat. It's like a very high average. But if you have not done well, just make sure that you do things accordingly, what you should do. I will not be able to tell that. But if you have done everything correctly and still having a problem, then talk to me. We will devise a plan. And just one thing that I like to mention, that throughout you have done very well, and sometimes if you're just in a borderline, say 91% or 90%, you are just like almost sure A, somehow you just did not do well in the last exam and it just dropped to 89.4. And remember that I teach such a large class. 89.5, I will round it up to an A. But if it's 89.44, I can't do that, because if I just do one, I have to do it all throughout in every course, every semester. Otherwise, it will be unethical. And I had to take that decision, and that's why it is mentioned in the syllabus. So just make sure that your course total just for the last one doesn't drop, so that you have a little bit of a buffer and the breathing room. And you will have, of course, the 4 quizzes as well. So that's just a note of caution, just to be proceeding with that way. All right, so let's start with the transformation oncogenesis, and probably you're going to see that this is more like a story. And a couple of slides, they're just a little bit intense, but overall it's pretty easy, and it's a fascinating story, probably the most fascinating out of all 17 chapters that you are going to see, that how virology paved a pathway for our understanding of cancer, basically. And a lot of people, if you just ask, many of the scientists even, that if you just think about the contribution of virology, I mean, like, it's a direct contribution of virology for our understanding of the cancer, and there is no question. It's like you are going to see how it is. So let's take a look. So first, before I start with this, I have posted a couple of videos from my cell biology lecture, just a couple of them. And they are really not long. So if you have some time, you can just take a look at over them, just like passively just seeing this, just go over that. It will give you a good idea that what we are discussing in the class. You do not have to really strongly memorize everything out of that, but will give you a good idea about what is a tumor suppressor gene, what is an oncogene, what are the differences, and it will just make this chapter much more understandable. They're at the very top, below the learning objectives as complementary, supplementary videos, a couple of them. All right, so the first thing what we are going to talk about, the primary cell culture and the transformation of the important cell lines. So basically, how do we make the cells? You can just take an embryo. Here is an example of a hamster embryo. You just trypsinize it, it's a protease, and it just disrupts all the cellular interactions into the single cell. You plate them, grow them. This is your primary cell culture. Why we call this primary cell culture? Because these cells are going to die just like the left, take a look at the graph on the left-hand side. They're going to grow for maybe 50 generations, maybe even 100 sometimes. But eventually, they're all going to die. But what you can do, that you can just put some UV, or you can put the mutagens in there. And after these many generations, many of them are going to die. And then suddenly you may find, again, the word may is very important. You may not find in 1,000 petri dish, but just one magic petri dish, you suddenly can see there is some growth. And this is what we call as a transformed cell line. And these are your immortal cell lines. And most of the cell lines that we used in the laboratory are immortal cell lines. And they are derived from a cancer tissue usually. But oncogenesis, cancer, and transformation are two different distinct events in a human being, and we are going to talk about that. Interestingly, what happened, mouse cells, if you just take primary cells and grow them in culture, they have the ability to transform spontaneously. On the other hand, if you take human cells or the primate cells, they don't until you put some mutagen or something in there. So you can even see that both vertebrates, rodents, vertebrate, primate, but just in between these two, they're very, very similar. We definitely do have an extra robust capacity to check our the cause of transformation, which are basically a series of mutations. I'm going to see that. All right, these are just very self-explanatory. That transform cell property, they're immortal, reduced requirement of serum growth factors, because what they do, that some of these cells produce their own growth factors. What we mean by that own growth factors, the growth factor genes are in the cells. All the cells doesn't produce it. Somebody else produces it, and when the cells get the signal, they will grow. These cells turn on those growth factor genes, and they secrete that and get stimulated, and that is what the reason that they do not require the serum growth factors. Loss of capacity to growth arrest upon nutrient deprivation, very low nutrient, they can grow. Growth with high saturation density and loss of contact inhibition. This is an interesting part that most of the cells, when you're growing on a Petri plate, if they touch each other, they will stop growing, whereas they can pile up on each other. And altered morphology, anchorage independence, some of them can just float and still survive, and they can be tumorigenic, benign or oncogenic. That is what the difference in between a transformation and oncogenesis. Just a growth of transformed cell does not mean that that cell is cancer, like in vivo in a body. What I mean by that, in the next slide, I will see. So here what we are looking at, that the tumor is a swelling which is caused by abnormal growth tissues. But again, originally it is a swelling, just not by the growth. Now, this could be benign or malignant. On the other hand, here you can see that just a picture of normal cells, abnormal cells, and then abnormal cells multiply. These cells are now transformed. Now, this cell can remain in here, just like think about a skin tag, and it can be benign. The cells are transformed, but they are not oncogenic. On the other hand, what happens that if there are some additional mutation, remember that benign mutations, transformed cells are not enough. It's a continuum. If they have more mutations by which they can invade other tissues and become oncogenic, that is your oncogenesis. So you need some additional mutation so that you can do that. So approximately about 12, but as low as 7. In between 7 to 12 mutations are usually required before you can have this cancer, basically. So a lot of mutations, a lot of things need to go wrong. And remember that all of these wrong things need to happen in one cell. If it happens in different cell, which happens every day, it really doesn't matter. But all of these wrong mutations have to happen in the DNA of a single cell, and that can become either transformative or more mutations oncogenic. So that's basically the concept. And mutations may be inherited, caused by the DNA damage, carcinogen, infectious agents, including the viruses. And also, interestingly, what happens that as we age, as the cells are being replicated, more they replicate, chance of having them more accumulation of more mutations increase. So think about that, primary cells divides 50 times. If your body, the cells, one cell divides that many times, it's going to accumulate those kind of mutations over the period of time. So these are some of the perspective, general perspective. So here what happens that you take the normal cells which are transformed, and if you just inject those transformed cells into a mice, some of them may not cause cancer, but some of them can cause cancer. That is the difference between a transformed cell, not oncogenic, versus a transformed and oncogenic cell. But one of the important things is that if you have a cell which is transformation, immortalized, oncogenetic, you have to inject it into a mice which doesn't have an immune system usually. If the mice actually does have an immune system, proper B cells and T cells activation, these cells will be killed by your cytotoxic T cells and the natural killer cells. So you have to see the tumor, the mice has to be immunocompromised. So what happens that studying the virus transformed cells provide the insight into the molecular events to establish the oncogenic potential, but no virus can do it all. So that's the important part. There has to be something wrong with the cell, and the virus can take the advantage of that. Just the virus is not enough to cause cancer. Any virus, no matter what, we're going to see that. So here, this is the very interesting part, that the cancer that is caused by the virus is just a secondary event. What it means that there is no way that the virus need to make the cell mutate in such a way that it causes cancer. It doesn't help the virus in any way. So the virus really, for its replication, for its propagation, doesn't need to induce cancer. It's just an event that happens as a secondary event. Except probably one. This is called a walleye dermal sarcoma virus. And this is where the virus evolved in such a way that, the sarcoma basically help the virus to replicate properly and propagate. And we're going to see that in a few slides, that what is happening in here. So this is probably the only unique example where the replication is helped by the dermal sarcoma formation. There is no other example except this. All right, so here are just a long list of different type of oncogenic viruses. And what we are going to see is like here we have this hepatitis C virus. We're not going to discuss detail about that because the mechanism is still being worked out. And usually what happens in this process that they hamper the signal transduction pathway or the apoptotic pathway, so they don't allow these cells to die even when they want to die because of the viral infection. And then those cells receive oxidative damage from the reactive oxygen species, so their DNA is damaged in those cells, which then gradually, and then still they cannot die. Because what happens that if you have a DNA damage, the cells should die, they still cannot die, and that can lead to the cancer as well. So we are not going to talk about that, detail about that. Then what we are talking about, this hepatitis B virus, we're not going to talk about that as well. And this is a hepatinavirus DNA virus. Here you can have some interesting question. Remember that this is the hepatinavirus do have a reverse transcriptase in its genome. Remember that this is a gapped DNA genome. It has a reverse transcriptase in its genome, in its life cycle as well. But it doesn't have an integrase right blood retroviruses. So in no way the hepatitis B virus can integrate its genome into the human beings, just like unlike other retroviruses. But there are some unfortunate events that sometimes the DNA fragments also can have rare mitotic recombinations that can happen. And those recombinations can lead to cancer. So we are not going to talk about that, but pretty much we are going to talk about pretty much the retroviruses right here, not specific one, but we're going to talk about the retroviruses, herpes viruses, polioma, and papilloma viruses in detail. So these are some of the things that basically happens with these viruses. We're going to talk about that. All right, so this is a very interesting, so this is the story of discovery of the viral oncogene and how it goes. So it is known as Peyton Rouse. He was a researcher in a Rockefeller Institute. And by that time, just one thing, just remember that here RSV is Rouse sarcoma virus it is not respiratory syncytial virus that you see that kids get RSV infection. This is Rous sarcoma virus. So pretty much what happened, the story goes like this. A farmer from New Jersey bring a chicken and told him it's like they have these tumors once in a while they find. And he told him, it's like, you are a scientist, you just find out that why this causes, what's the reason for this tumor. And he just got interested in that. And that was basically the idea. And he found that that tumor is caused by the Rous sarcoma virus. And it's a very interesting thing. At that time, when he told the scientific community for that, nobody believed him. This is the Rous sarcoma, 1990, nobody believed him that the virus can cause a cancer. It took 50 years for the scientific community to understand the implication, and he received the Nobel Prize for the discovery of 1909 in 1966. But that is something, and how it is so is that, think about that, after that Harold Varmus and Bishop, they identified the first oncogene, and this is a viral circ. The SRC is the SRC, it's not pronounced as SRC, but it's pronounced as a circ, which comes from the sarcoma, basically. They found the first time that in the RSV, there is a circ oncogene in the virus, integrated into the viral retroviral genome, and that basically was the first identified oncogene, and they received the Nobel Prize for that immediately for that. And after that, it was like people discovered many oncogenes. Like it was just like opened up a whole new door, basically after that. On the other hand, what happened along with that, this is the interesting thing. Then until 1950s, the RSV can cause cancer only in live chicken. But what Howard Temin, and you have known that Howard Temin, so this is Renato Dalbecco, who did the plaque acid. And Howard Temin was a graduate student in Dalbecco's lab. And he was working with the cell culture and those. And when it was found that RSV can cause cancer, he was working on the Petri plates growing cells in culture and thought that if they're infecting with the RSV, he can do the cancer on a Petri plate, basically replicate the disease. It took him 7-8 years constant effort. I mean, like how many times you have a failure and still think that one day you will be able to do it. But he did that. So now you have the transformed cells by the RSV, which he made in the Petri plate, completely changed the whole scenario that how we study virology and the cancer causing thing. And you all also know that he is a co-discoverer of the reverse transcriptase along with David Baltimore, and both of them got the 1975 Nobel Prize. So he worked for a long time in his career at the University of Wisconsin and Madison, and they have an extremely very big virology department. And there's a big lake in there called Lake Mendota. If you ever go in there, you will see. And there's a long lakeshore path which is named after him as the Howard Temin Lakeshore Path. That was a picture. Another interesting thing is that, which is just a little bit out of the way, that this is one of my high school friends. We just went from elementary, middle, high school. And then he got his MD and he got his PhD from the Albert Einstein School of Medicine in New York. Worked as an NIH, and now he moved to Bristol Myers Squibb. So he's the head of the their oncogenic products or development in there. And he was working with Harold Varmus in the early 2000s, and he was working on the receptor tyrosine kinase signaling lung cancer with the Varmus. So think about that, 1970s, he was working with discovering the oncogenes, and he's still doing exactly the same thing. Remember that Harold Varmus was the director of the NIH for a long time and the advisor for the president. So big scientist, big name. So that's very interesting. So this is the reason that we say that virology paved the pathway for our understanding towards cancer. So let's take a look at how the Varmus and the Bishop found the oncogene from the RSV. And pretty much a very elegant, but a pretty much straightforward experiment. It's called a subtractive hybridization. So what they did, think about this. You have the RSV genomic RNA, and then they reverse transcribed that RSV. Reverse transcriptase already discovered by that time. So they took the reverse transcriptase, RT, that you use in your RT-PCR as well. So they reverse transcribed those, and then the fragments, they just chewed these fragments with some endonucleases, so they are fragmented. Now, they have also found the RSV, that there are some transforming RSVs that they can find from the tumors, but there are some non-transforming RSVs. These RSV, or the Rous sarcoma virus, don't cause tumor. Even if you inject them to the chicken, they don't cause a tumor. So these are non-tumorigenic. So what they did, that they took all these probes and hybridized with it. And they found, and then after hybridization, they just passed that through a hydroxypeptide column. So all these hybridized probe basically just got stuck, larger ones on the hydroxypeptide column, whereas the probes which did not hybridize with the non-transforming RSV genome, RNA genome, they came out. And they found the sequence from these, and they found that there is a SERC oncogene in there. So remember that the SERC gene is a human gene, and that was in the viral sequence, that was in the virus itself. So they basically argued and then chalked out the pathway that this must be an oncogene because this is not a viral gene. So the virus got it somehow from the cells which they infect. So these are just, on the other hand, essentially the discovery of Howard Temin, that how you can transform the cells basically in pictures. So this is the foci which is getting transformed and how they change over the period of time. Just a very self-explanatory idea. So how can a viral infection transform the cells, and what are the requirements? So the first thing is that the cytopathic effects must be reduced or eliminated. If the cell is killed by the viral infection, it's not going to get transformed. So you have to keep the cells alive. That's number one. Then viral replication also must be reduced or eliminated, and the transformed cells do not produce virions. Because think about that if the cell is constantly producing virions, at a certain point of time, the immune system is going to attack and kill the cells. If the cell doesn't produce any virion, then it will escape from the, eventually escape, approximately for a certain period of time, will escape from the immunosurveillance. And the cells must continue to divide, and it becomes immortal. Sometimes the cell can understand that there is a viral replication and tries to make the apoptotic or programmed cell death, but in this case, the cell cannot die. It cannot do programmed cell death, so basically the apoptotic signals are completely blocked, and these are the cells which become immortal. Now let's take a look at this walleye dermal sarcoma virus and its unique transformation cycle. So if you just think about that, how this retrovirus genome sequence is, you already know that they have LTRs, very, very active promoters on each side. And this GAG pol on the envelope, these are essentially the core sequences that every retroviruses have. In addition to those, here you can see that there are some accessory gene sequences which produce accessory proteins A, B, and C. These are not found in all of them. This is very specific. And it was found that these A and the Bs, they produce different proteins, and how they basically control the walleye dermal sarcoma formation is extremely unique. So what happens that during the spring, when the water temperature is actually high, at that time these tumors, they start growing. But after growing those, they actually do fall off very quickly. And the virus can transmit to others. And this is a process of transmission. They don't grow as big that we saw before. So we saw right here This is a bigger one. They do not grow that big. They're small. But what happens that during the fall, when the temperature of the water goes down, these sarcoma, they starts growing bigger and they don't fall off. So over the fall and the winter, when there is no spawning, there is no reproductive event of the fish, these basically grow. And in the spring, when the water temperature rise, they basically fall off. So basically, that will help the virus transmission. So how it is basically done? It is found that these ORF A and the B gene, they produce some very specific proteins which promote the cancer, basically. And so these are activated in the fall when the sarcoma is basically growing. Now what happens, the C is an inhibitor of these proteins. And the C is activated when the water temperature rises, basically. They code for a cyclin. I put the detail in the notes. But during the summer, during the spring, when the water temperature rises, the C is activated. So C is temperature-dependent activation of that. When the C or C accessory protein is produced, that inhibits the oncogenic A and the B, and then the tumor falls off. It doesn't grow anymore. So this is the mechanism that with a very unique cycle. All right. So after that, what we are trying to do, that the route to understanding the viral transformation in the culture and the relationship. So basically, what happened that initially, this is 1950s. People used to think that okay, all right, the RNA viruses or the retroviruses cause cancer. In the 1920s, people used to say that the DNA viruses cause cancer. Studies with the polyomavirus can cause cancer. And 1940s, the cell biologists told that it is a cellular phenomena. But all of them fought against each other. They thought that their process is the process by which the cancer happens. It was not until 1960s and the 70s that the unified theory of cell growth control, which is a present theory, all converged. That basically all of them, no matter whether it's a DNA virus or an RNA virus or a retrovirus or the cellular phenomena, all of them basically employ the same mechanism for cancer. And that unified theory is, we are going to see that how this happens. So this is, these are two examples, and we're going to see that this is the first part, that how, so this is the first part, is that how the retroviruses cause cancer. This is what we're going to talk first. So this is the start of that retrovirus which can cause cancer. So first, the RSV cause solid tumor sarcoma. Viral isolates from the tumor cause rapid sarcomas, means that they form the solid tumors, not like leukocysts or the, so basically the leukocysts or leukemia, whatever you could call me, that means that you have an abnormal growth of the leukocytes, white blood cells, and we call them blood cancer, leukemia, or leukocysts, any of those. That is not what it is. This is like a solid tumor formation. And one of the things that here, this could be a little bit confusing here, most of these viruses are defective. And we're going to explain that, what do you mean by that? And the viral genomes from the solid tumors are recombinants. So what these two things mean, we'll come back to this in a second. Let's take a look at another virus, which is the avian leukemia virus, retrovirus as well. This is an endemic in a chicken. And initially, when a chicken is infected, there is a viremia. That means that there is a replicating virus into those. And the chicken become, almost 97% of them just become immune to it. And the older birds, remember that this is the word, older birds, develop sporadic leukemia and variety of tumors, but it's only 3%. And most of these viruses are defective and all had a different host DNA. So what we mean by defective and contain host DNA and recombinants. It was very interesting that was found, And this is one of the reasons that Peyton Rouse was lucky. When he isolated the RSV that he was growing, that virus was not defective. That virus was not defective and had an oncogene. So that's why he could grow that virus in the cell culture. On the other hand, what we are talking about, that the defective viruses, these viruses can replicate in the cell if only helped by a helper virus. So we will explain that, what is meant by the helper virus. So think about this, just the analogy. 42% of our genome has retroviral sequences. So in a cell, you have a retroviral sequence and several one of them. One is a defective with an oncogene. That is trying to make the virus, but it is cannot make a full virus because it doesn't have all the GAG, Pol, and the envelope gene. But another sequence can express the GAG or the Pol or the envelope which is missing in this virus. And if they're replicating at the same time in the cell, the defective virus still can grow. And that is the unique thing, that how the defective viruses can also cause cancer, but the probability is less. And it will just become a little bit clear in a second. So I'll explain that what we mean by the defective ones and not defective ones. So here we are looking at that how the retroviruses transform cells by 3 mechanisms. One of them is a rapid tumor formation. RSV, about 2 weeks. And the RSV has an activated dominant oncogene in its genome, like a VSARC, and the protein, oncogenic protein, is produced immediately when the virus replicates. This is basically happens in a non-defective virus. On the other hand, what happens, so that's very important. Intermediate kinetics of the example for the avian leukemia virus, it takes months. The ALV carries no dominant V-onc or the oncogene, ONC for oncogene, that's not a specific name, this is generic. And it causes cis activation. The provirus turn on expression of endogenous oncogenes. What we mean by that, remember that all of these retroviruses are going to insert their genome into the DNA. Somehow, if it enters or inserts its DNA close to a proto-oncogene, which can cause cancer in the human beings, and if the LTR of the retrovirus is extremely powerful promoter, drives overexpression of this proto-oncogene in the primate or in the host, that can cause cancer. So here, cellular gene, integrated retrovirus, retrovirus is causing a cellular gene to overexpress to cause cancer. That probability is low. That's the second case. This is because this is a chance event. And the third one we're not going to talk about much more. The HTLV carries no dominant oncogene, does not cause a cis activation of the local oncogene. Viral regulatory protein activates the oncogene by a trans activation. So this trans activation, we talked about that this is an activation of an oncogene far, far away, maybe thousands of base pair away. And this basically needs a DNA bending and activation of that. Chances of that is even less. Cis activation means, okay, all right, you have a viral sequence next to an oncogene. Increase that. Here, no oncogene nearby. It's very far. But a trans activation by bending the DNA can cause the cancer, and very slow kinetics. And we're going to look at them in the pictures as well. Before we look at the pictures, we will come back to this slide back again. These are the genomes of the transducing retroviruses. So let's take a look at what we are looking at. Again, you do not have to memorize any of the names. But here we are looking at an avian leukosis virus, generic. You have the GAG, Pol on the envelope, and of course you will have these green ones are the LTRs at each end. Now, this is the Rouse sarcoma virus. You can see that the Rouse sarcoma virus, which Rouse basically isolated the one which was not defective. Which was not defective is this one. So what he found that you have a GAG, you have a Pol envelope, and you have the SERC oncogene inserted into the virus. So the virus
could package a whole sequence of the VSERC oncogene into the virus. So the SERC oncogene intact, along with some additional sequences to express it, is in the virus. On the other hand, if you just look at the other one, lots of different things, see that out of this pink, blue, and the brown color, something is not correct and something is missing. So here you can see that right here, avian myeloblastosis, GAG is good, POL is good. See that the envelope is pretty much deleted, so the MIB gene is inserted into the envelope. See that the brown in here. See right here the blue POL is deleted. So that's basically what it is. So these are sometimes what can happen that instead of just one oncogene, some of them can have even two oncogenes. And these are all defective, but they can all grow with the helper virus in the cell. So that's one of the things. Here are the mammalian transducing virus. The same thing is like a GAG-POL envelope. Murine is a mouse leukemia virus. And you can see that right here, these are the ones. And most of them are defective viruses. And that's one of the reasons that when you have a defective virus, the chance of having the tumor is much less. If you have a virus like this, Rous sarcoma virus, with an intact gene or the intact sequence of the whole virus, along with an oncogene, the infection will immediately cause cancer. So that was what we are looking at. But all these ones, intermediate and the slow kinetics, are these which are all defective retroviruses. It takes months and a helper virus, and it's a chance event. So this is the mechanism of oncogene capture, that how they get these oncogenes, basically. So very interesting that how it gets. Right here, so think about that. This, you have the viral DNA, GAG, POL, and the envelope integrated into the host DNA just right here. So you have LTR, GAG, POL, envelope, and the LTR. So this is your retroviral sequence integrated in the DNA. Somehow think about that you have an oncogene close to it right here. So these are the oncogenes, and in between that, those you have the intron sequences. So you have a wild type mRNA, and then from here, you are just making this wild type mRNA, and sometimes what can happen is that as they are making these mRNAs, sometimes there is also the deletion of the virus and the cell sequence, and they package these oncogenic sequence along with the virus particle. See that, or try to. So this is being produced in the cell. But what is happening that this is defective. So how this can happen? Because this yellow thing is the packaging signal. Without that, this will not be packaged in the thing. So remember that this is where the copy choice mechanism helped the virus. So remember that each of these retroviruses have two copies, or supposed to have two identical copies of their genome, both of them the same. Here, they are not the same, and here there's a copy choice mechanism. So what is happening that the virus is trying to replicate this genome sequence from here, then switches, and this is just completely random, takes this sequence and ends it up here. So the recombinant one will have the packaging signal from here, packaging signal from here, and this is how they captured the oncogene. So that's basically one of the mechanisms. On the other hand, the other part that also works is sometimes when the viral the mRNA is being made, which is also the genome, there is a read-through through the LTR. So it starts from here, the mRNA is made from here, and it should stop at the end of the envelope. But sometimes what happens during the transcription process, it doesn't end at the envelope, and it just goes all the way towards copying the oncogene. And of course, the introns will be removed, and that part is packaged into the virus like this. And the same way, by this copy choice mechanism, because remember that you have to have these two packaging signals in both. This packaging signals basically starts from here, picks up this oncogene, and just goes in here. And now what happens, this can be integrated into the host back again. So remember that this is happening in the cell, and it doesn't need to go and infect somebody else. This is just going to get integrated into the cell back again, and that's basically the process. Now you have the oncogene, and sometimes on top of that, what happened, that there could be rearrangement, duplication, point mutations, and all these happens because remember that we have these retrotransposons in our genome. That's a normal thing. These elements move around in different ways. That's a normal thing, and by that process, they can be inserted in wrong places, wrong time, and that can lead to the cancer. So first, we are going to talk about the proto-oncogenes, what we mean by the proto-oncogenes. The proto-oncogenes, and there is no gene in the cell that is there for causing the cancer. But what we mean by the proto-oncogenes, that these are all cell cycle control genes, basically. There are more than 60, extremely highly regulated. And the cellular genes are abbreviated as C-oncogenes, ONCs, this is your generic name, but SERC, MYC, MOS, RAS, these are the specific oncogenes, or proto-oncogenes. So on the other hand, if you have a C, that is a cellular SERC, cellular MYC, cellular MOS, cellular RAS, retroviruses isolated from the tumor have altered copies of the C-oncs, and they are viral oncogenes, VSERC, viral SERC, viral MYC, viral MOS, viral RAS. That's the basic difference. So what these proto-oncogenes basically do, these proto-oncogenes, when expressed, make the cell divide. So they are off all the time. They're off all the time. That means that they are highly regulated off all the time. If not necessary or the cell is not ready, it will not allow the cell to divide. On the other hand, what happens if you just express them and they will just turn on, in that case, they will divide the cell in an uncontrolled way. So this is one of the type, that is the proto-oncogenes, and we're going to see another type as well. Now you have the connection between this picture, retroviruses transformed by 3 mechanisms. Now you have a picture of this. Transducing is the rapid. So think about that. You have an LTR. You have the LTR. Of course, you will have the GAG-POL and the envelope gene. And the V oncogene is right there inside the virus. This is a rapid transformation, like the non-defective RSV. This is a cis activation, which takes months, maybe years as well. Here you have the GAG-POL envelope of the retrovirus, which inserts itself close to a cellular oncogene. And sometimes, remember that, this LTR is an extremely powerful promoter, and sometimes this can enhance the expression of the C-onc. And what we said right here, these are usually off, highly regulated. When they are elevated in their expression, they tell the cell to go into cell cycle or continue the cell cycle or to divide. So when you express this, that will tell the cell to divide. That's the mechanism. On the other hand, the transactivation, just think about that this is your the retroviral sequence, inserted retroviral sequence in the DNA. And this cellular gene, say a IL-2 or IL-2 receptor, this is far, far away, nowhere close. That's why it's separate, nowhere close in here. And the viral transcription activator, basically the LTR, just by the DNA bending can activate the promoter thousands and thousands of base pair away. And this is basically the transactivation. Chances of this is even low. So you will probably thinking that what IL-2 and the IL-2 receptor is going to do. Remember that those are cytokines. They will tell the B cells to divide. So that uncontrolled B cell growth will just basically can cause the leukemia as well, or all of the leukocytes are some kind of immune cell uncontrolled growth of the leukocytes, basically. So this is the byproduct of their life cycle completely. This is not that what the virus wants to do, though. So the next part, what we are looking at, that this is just an insertional activation of non-transducing oncogenic retroviruses. This is just an example of that, just one. And again, there's nothing that you have to memorize from here. Say for the avian leukemia virus, you can see that this is the c-MYC gene, exon 1, 2, and 3. These are the different insertional sites where the ALV just gets its gene. So basically, that's what it is. These are the different ones. These are the directional way that where they are from, or the transcriptional way. So these are the insertional sites. So here is one example of this promoter and the enhancer activation, basically. So here what is happening right here is that this is the cellular oncogene, remember that. And they also have these exons and the introns in here. So here what we are looking at, that you have the genome of the retrovirus, you have the LTR-GAG-Pol envelope retrovirus. Here you have the C oncogene, here you have the C oncogene. So it is inserted somewhere here. And the LTRs, sometimes what happens, that drive the expression of these, and sometimes these overexpression of these can cause the overactivation of the oncogene, basically. So here in this case, what happens, that there is a deletion at the left end of the virus, basically. So you have this oncogene C on this side, but you don't have this C oncogene on the other side. So you are just making this this protein, and sometimes these, even in a defective form, that can be enhance the cell cycle. On the other hand, there is an insertion, that what you are looking at, that you have an LTR, and here the oncogene is right here. And that is basically, on the other hand, what happens, that either it can be upstream or the downstream, but this is a trans activation, whereas this is a cis activation that we have seen. You have an oncogene right here, and the virus is right here, which is just only the LTR has been shown, and that is driving the transcription of the oncogene. Here there is an LTR, and the virus is on this side. Here the virus is on this side, here the virus is on this side, but this LTR can trans-activate the promoter of the oncogene, and that can also overexpress these proto-oncogenes. So these are some of the activation of non-transducing oncogenic retroviruses. None of them has the intact oncogene inside the virus, but they are close to something or defective. So now that's all about your retroviruses. Now let's take a look at the 1920s, the DNA viruses. So the DNA viruses are the first oncogenic DNA virus. It was a papillomavirus that was caused the worst papillomas in cottontail rabbit, and Richard Shope in 1933 discovered that. And here you are looking at some of these structures. These are some of the growth, abnormal growth from their epidermis due to the papilloma. And this is probably, look up this, and these are probably due to this. They are called a jackalope. It's a mythical creature. And probably people saw these in the wild and thought that these are the jackalopes. And these are nothing but this growth by the papillomaviruses. And it really sometimes, most of the times, doesn't kill or doesn't do any harm to the rabbit. They just fall off, just like the antlers as well. So a very interesting discovery. And then Ludwig Gross discovered a murine polyomavirus in the 1953s. And then also it was found that pox viruses can also cause fibroma in the animals. Very, very rare, but it can cause. And remember that the first thing is that the DNA tumor viruses, very rare transformation and tumorigenic potential. So we have seen that if the most potential is a retrovirus with an oncogene in it is the most potential. If it's a defective, less. DNA tumor virus, even less. Very less chances of a DNA tumor virus will cause a cancer. And we are going to see that why it is. So one of them is adenovirus. Many serotypes do not cause cancer in the humans. And they're 12 to 18, they can cause in hamsters. Adenovirus 711, poorly tumorigenic in the hamsters. And the tumor transformation cells like the polyomaviruses, papillomavirus, and they are very rare events. So we are going to see that why this basically happens. Remember that. Doesn't cause in the human, but in the hamsters. And that is very important, hamsters or mice. So next, what we are finding that out of the DNA viruses, and this will be your, right here, the polyomaviruses, and SV40. And we have talked about this SV40 LT for a long time. And now you know that right here is LT, large T antigen. We did not define what is T. It is a large tumor antigen. So that is where the T comes from. And what it is that there is a large T, large tumor antigen, and a small t as well in the polyoma, papilloma, HPV-16 type. They encode this E7 gene, and adenovirus produced a T antigen or the tumor antigen E1A. All are different proteins. So what are the tumor antigens in some of these? So just remember that polyoma has the LT, E7 in papillomavirus, E1A in your adenovirus. These are the ones which are the key. And it was very interesting that it was found that they do all share some cellular sequences of some of the cellular proteins. And we're going to see that why that helps. Remember that all of these antigens do have a cellular sequence in somewhere in them. And this is how the virus basically evolved. And let's take a look at that, how this happens. So 3 seemingly unconnected discoveries that we are talking about, and understanding the link between the virus's transformation in the cell cycle. So a 53 kilodalton protein binds to the SV40T antigen. And then what we found, there's another entirely different event. Transcription of a set of adenovirus early genes, the E2 gene cluster, require a cell protein, E2F, the E2 factor. Now a family of proteins called the E2F family. And the E2F found to bind a cellular protein called the retinoblastoma or RB. All different one. And this is the most interesting part. That 53 kilodalton cell protein binds to SV40 T antigen, and that is your P53, which we call as a guardian of the angel. The guardian of the, not the angel, not guardian angel, the guardian of the genome. Basically, if there is any DNA damage in the cell by any reason, P53 is basically the protein that is induced and cause checkpoint or the arrest of the growth rate. And we are going to see that how the E2F, on the other hand, basically controls the RB or the retinoblastoma that we already talked about this. And basically, the P53 RB E2F subsequently discovered to be critical players of the control of the cell cycle. And it was all discovered through the virology. So here, what we are looking at, that this is the overview of the cell cycle again, that if the conditions are done right, the cell cycle pauses at a restriction point. And if we just look at the cell cycle has a G1, growth 1, then you have a restriction point right here, and from there it goes to the S or the synthesis phase. DNA replicates here, then G2 phase, the second growth period, and this is your mitosis basically. So what happens, the protein that regulates the restriction point right here is RB, retinoblastoma protein. Now, if both copies of the RB gene is lost, you just the kids develop the the retinal tumors from the retinoblast, not as an adult, because these cells in the retina, they are gone by the age of, or retinoblast, which makes the retinal cells, rods and cones. They're gone by the age of 5. You have all mature rods and cones. But what is important is that RB is a recessive oncogene, and it's a tumor suppressor gene. So far that we talked about the retroviruses, they were making a gene, a proto-oncogene to an oncogene. That means that if you just have that protein, normally it's not expressed. Expressed, cause cancer. Tumor suppressor gene is produced all the time, which controls the restriction point. If you somehow inactivate the tumor suppressor gene, it will kick the cell cycle into division, and that is basically the mechanism of the tumor suppressor gene. Produced all the time, you're turning it off, exactly opposite to the oncogenes. So here is a process that how it happens. So here is a receptor tyrosine kinase signaling, and what happens, that when there is a signal or a growth factor basically binds to this, say epidermal growth factor or anything, it just does a lot of signaling. And you do not have to remember any of these. You learned this in cell biology, but not here. And I'm not going to ask any of these. So once this signal transduction pathway just goes in, the final product is that activate this cyclin and CDK4. So see that this is CDC4, is not 416. This is 4/6. So it's either cyclin D1 CDK. This is not even CDC. This is actually CDK. I hope that I corrected that in the writing. So this is a cyclin, and this is a cyclin-dependent kinase. So this is CDK4 or CDK6. Once these are activated, so it's a kinase, it phosphorylates the RB. If the RB is phosphorylated, it is now inactivated. Now the cell can basically divide. And we are going to look at the mechanism that how this basically happens. So take a look at the whole mechanism. You have seen this picture before in a different context, but we are going to see this in a little bit more different way. So let's take a look at this first, right? Here we are starting from here. Here you have an RB produced all the time in the cell. It binds to a transcription factor called E2F. And you have an E2F dimerization partner binding to that. So what they do, that this complex, when the RB is binding to this, basically binds to the histone deacetylases, these HDACs, histone deacetylases. Remember that if you need the transcription properly, you have to make the DNA loose. So you have to deacetylate DNA. You have to remove the acetyl group from the DNA. But if it binds to the histone deacetylases, it cannot do that. So the DNA is compact because the acetyl group is still there. So no transcription. On the other hand, what happened? See the cyclin D CDK4/6. So this is correct. So see that cyclin D right here, and then CDK4/6, and cyclin E CDK2, which is not shown here. These kinases right here, cyclin dependent kinases, cyclin E dependent kinases, they basically what they do, make the RB phosphorylated in several sites. So this is basically the process. Now what happens that when it phosphorylates this right here is the E2F and the DP. They are basically free, and it will make also the HDAC free, so it can uncompact the DNA, and basically the transcription can go on. Now here what we are looking at, that as the transcription is going on, so right here as transcription is going on, it also feedbacks into the loop. It produces more cyclin D, cyclin 2 CDKE, cyclin 2 EF. Basically these are the proteins which even help in more of a cell growth or the DNA synthesis. So this is a positive autoregulatory loop. On the other hand, the process is very controlled. There is also another protein called INC4, which can have a negative effect if the cell is making enough DNA and enough cell growth is going on. If they need to shut it down, there is also an inhibitory process. So you have a positive autoregulatory control, negative autoregulatory control, very well controlled. Now this is the normal process. Here you can also see that a cyclin A CDK2 that also help in the anaphase promoting complex, includes cyclin B, it drives the mitosis. So remember that this is basically the S phase or the synthesis phase of the cell cycle, and this is the M phase of the cell cycle, and all is controlled. So remember that restriction point, we turned turned off the RB, inactivated the RB, it goes into the synthesis phase through G2, it will go to the M phase, which is basically cell division. That's what we talked about here. Now see the top portion only. E1A, that's your adenovirus protein. LT from papilloma. E7 with E1A adenovirus, LT from the SV, E7 is from the papilloma. All of these are the viruses implicated in the human cancer. What basically they do, that right here, they bind to the RB just like the phosphorylated portion of the RB. So that's basically the thing. that right here, you can easily see that as they bind right here, as they bind to the RB, the effect is the same. And all of them, E1A, LT, or E7, they do not phosphorylate, but they bind to a site. So that's why I said that E1A, LT, E7, all of them has a sequence which can bind to the protein in the cell, which is RB. And these are your histone deacetylases, which will activate along with the E2F. And remember that as this binds to the RB, E2F is DP is free. So this is driving this. Normally in the cell, this process is done by the cyclins and the CDKs, which phosphorylate the RB and make these transcription factor free. Here, these are making the transcription factor free and also will recruit the histone deacetylases. And basically, DNA synthesis, mitosis, which is the cell division, basically. So basically, the T antigens, remember that the tumor antigens, although only LT is known as the large tumor antigen, but remember that your E1A, E7, these are considered as tumor antigens as well. It's just a different name. All of them are tumor antigens. And the T antigens kick the quiescent cells into the S phase and also inactivate the P53. So how this one happens. So here you can see right here, this is the same picture. E1A, LT, E7, these are the T antigens. They are just kicking them, the cell into the cell cycle. Now, the entry in the S decision is under more control. So this is a way that the virus needs to find the second impediment. So here, this is done by the P53. If the cell is not ready, the DNA is damaged or there is something in there, P53 helps that or controls the restriction point as well. It will not allow, even if the RB is inactivated by the E1A, LT, or E7, still P53 can control or checkpoint. This is also countered, and this is what we're going to see. So let's take a look at how the mechanism works, that in the cell, this MDM2 is produced, this is a ubiquitin ligase, and P53 is produced. As soon as the P53 is produced in the cell, the MDM2, which is an ubiquitin ligase, basically adds this ubiquitin and they are degraded by the proteasome. So in the cell, P53 is produced constantly, but it is extremely unstable, means they are degraded within 30 minutes. They're produced, degraded, and this degradation is done by the MDM2. And what happens is that this ARF, we're not talking about that, this ARF is a negative controller of that, which is also activated when there is a DNA damage, this ARF. This can, when it binds to MDM, it will not be able to degrade the P53. And the ARF is signaled when there is some damage. But let's take a look at from here. So this is the process that we said that degradation happens. But now we are starting from here. Suppose there is a double-stranded DNA break. This basically is constantly scanned. The DNA is scanned by a protein called the ATM. There is another protein called ATR. And these are all implicated in ataxia-related proteins. And then they have this checkpoint kinase. These are the checkpoint kinase, or these c-Abl. All of them phosphorylate P53 in some specific residues, phosphorylation. So these are checkpoint kinase phosphorylating. As soon as they do this, see the T half is now increased from 30 minutes to more than 2 hours. So the P53 is stable at this time. So what P53 does at this time, that's the P53 basically recruit a very potent cellular transcription activator called P300 CBP. The cyclic AMP binding protein. And this right here, the tetrameric P53, along with the cellular P300 CBP, is an extremely powerful transcriptional complex. This basically gives rise to many different types of protein like P21 CYP, BAX, MDM2, more of these. And these proteins basically cause the cell cycle arrest, and the BAX also called apoptosis. So basically you have a DNA damage. These basically check the cell cycle so that it cannot go on, and also promotes the cell killing if it is needed by the programmed cell death, so that the cell, if there is a damage, doesn't happen. So this is basically the process that how p53 checks the cells with damaged DNA in this process by enhancing the transcription. Now, let's take a look that the viral proteins basically inactivate the p53 and how it is done. Again, much very complex slide, but that is really not what is needed. So right here, think about that adenovirus papilloma polyomavirus infection, you have a p53. Just know the mechanisms. One of these right here, what happens is like right here is the E1B, and take a look at this E4ORF or E1B. These are the proteins from the adenovirus. These recruit an ubiquitin ligase. So all of these that what you are looking, they are together ubiquitin ligase. As soon as the p53 is tagged with the ubiquitin, they are destined for the proteasome for the degradation. So no control of the checkpoint arrest or no apoptosis. On the other hand, right here, you can see that this is your E6, which is HPV16 or HPV18 E6 protein. The same way, it recruits ubiquitin, degrades. On the other hand, SV40, see that this protein does everything. It even binds to RB. Now it binds to p53 as well. So basically, it sequesters p53. That means that when it binds or to the p53, it's taken away from the cell. It cannot bind with the CBP300, or it cannot go onto the promoter to activate the transcription because they are not available. They are sequestered by the SV40LT. On the same way, the E1B 55 kilodalton protein, this is E1B, also sequester the blocks basically the increase of specific cellular genes. So basically what you are seeing that out of all degradation, sequestering, repression of the transcription binding to the apoptosis inducing proteins, so it checks the apoptosis, cell cycle arrest by this P21 cycling inhibitor protein, that cell cycle arrest, that is gone, and BAX APF1, these are also apoptotic proteins. These are not expressed because P53 is basically degraded. So that's how the viral, you can see adenovirus E4, E1B, human papillomavirus 16.8E6, SV40LT, adenovirus E1B, all of them basically inactivate P53. Along with the RB, the cell cycle goes on. So why are all virus genes basically, except T antigens encoding genes, deleted or turned off in SV40 polyoma adenovirus transformed cells? And why is transformation of these viruses are so rare? Very interestingly, what is found, the virus which causes this cancer, when you isolate them from the tissues, they are all defective. So what happens that these are some of the reasons. Number one, the lethal late genes right here, late genes must not be expressed. And this is by a rare spontaneous deletion of the late genes. And also this can happen in infection of a semi-permissive cells where the late gene expression is blocked. Say for example, a human SV40 trying to infect a hamster or a mouse, different host, and that can cause repression of the L. So LT is produced, but not these, which can kill the cells. As these are not expressed, the cells are living, and LT is doing its job, basically to move the cell cycle into uncontrolled cell division, that's growth. And the second part is that the T antigen must be on constitutively and transmitted to every cell, which could be all of these proteins. Remember that all of these proteins that we talked about right here, all of them are T antigen, tumor antigen basically. So that's basically the viral encoding T antigen must be integrated into the host DNA, which is not an easy job. You have to have a mitotic recombination where the LT from the virus needs to be integrated by homologous, non-homologous recombination, which is a very rare event. Both of them need to happen. Very, very rare, and that's the reason that the transformation or oncogenesis by DNA viruses are very, very rare. So here also, that the transformation tumor formation abnormal epigenetic process for the DNA tumor viruses, and they were all discovered by infecting the wrong host. So a human adenovirus when growing in a hamster or the mouse, and now we understand that even in natural hosts, these rare events may occur leading to the tumor genesis like the human papillomaviruses. And this, the very, very interesting thing is that now if you take the HeLa cell line, that was from the cervical tumor of Henrietta Lacks, you can easily see that the HPV E7 is sitting right there, and that was the oncogene. That's from the cell line. You can just easily find it out easily that which oncogene is causing that. And the DNA tumor virus must start the cell synthetic machinery to make the DNA. This is the summary of that. T antigens turn on the cell cycles from G1 to S phase through inactivation of the normal inhibitor RB. In addition to that, inactivation of P53 blocks the apoptosis, where the cells are basically not ready for the division. It basically clicks them. And if the lytic, which is basically killing the cells, lysing the cells, are blocked, cells making T antigens continue to divide, they are basically transformed. They are on their way to become cancer cells, which is a secondary effect that arise but does not causally influence a process. So remember that this process will make the cell divide continuously, but not yet they are cancerous or immortal in that sense. Or no, I'll take the board. They're immortal, but not cancerous yet. They cannot invade other tissues. Some secondary effect can happen in some cases where they can invade the other tissues and become cancerous. So it's a whole continuum. And then finally, what you have is that this is the primary two mechanisms. And very interestingly, and understand this very well, that the retroviruses turn on constitutively off proto-oncogene. Revealed by studying the retroviruses, these are the dominant oncogenes. On the other hand, DNA viruses turn off constitutively on tumor suppressor gene like RB. And these are studied by the DNA tumor viruses, and these are the recessive oncogenes. Remember that when you are turning on, just out of these two alleles that you have in your two chromosomes, if there is just one of them is a dominant, you just turn it on and it's going to just cause that. But here, recessive oncogenes, that remember that here what is happening, that DNA viruses turn off constitutively on. So that means that both of the alleles, if there is both of them need, like in a normal case, whether you have in a normal cell, if it is not even infected, both of the alleles must be recessive so that the protein is not expressed. That is a normal way to do the cancer. If one of them is active, it's going to produce the protein. So the virus is basically counteracting that, and these are the recessive oncogenes, which are the tumor suppressor genes. So that's it, pretty much, I guess. So in the next class, we will do the vaccine, probably one of the most anticipated thing. And I'm just waiting a little bit. I will open up the exam for the review. If you have any questions, just let me know, and I will just post an afterthought for the whole class as well. But if you have any questions, please let me know.